sebelah sana ya. Saya bagian depan isi dulu. Ya, segera kita mulai. Maju depan sini. Terus tambah sekali.
Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu, Namo Buddhaya, Salam Kebajikan. Good afternoon, everyone. It is my pleasure to welcome you all to today's general lecture, proudly presented by the Biotechnology Study Program in collaboration with Research Collaboration Center for Thermophilic Enzymes, which titled Thermophile Microorganisms, Exploration and Their Diversity. So first things first, I would like to extend a special welcome to today's speaker, the Honorable Professor Mario Konosawa from Department of Science and Engineering for Sustainable Innovation, Faculty of Science and Engineering, Soka University, Japan. Welcome to Semarang, sir, and hope you enjoy your stay here. And also, I would like to address the Honorable Dr. Sri Pujianto SSC MSc as the head of the Biotechnology Study Program. Welcome, sir. And also Dr. Renat Artobliharjo SSc Biotech as the head of the Research Collaboration Center for Thermophilic Enzymes, and also today's moderator. I would like to welcome all esteemed lecturers of Biotechnology and also Biology Study Program, and of course all participants, students who joining us here today. Very warm welcome to all of you, ladies and gentlemen. My name is Aurora, and today I am very honored to be the master of ceremony for this special event. Today we will explore the fascinating world of thermophilic microorganisms with our distinguished guest, Prof. Mario Konosawa from Soka University, Japan. So to give you an overview of today's agenda, we will start by singing our national anthem, Indonesia Raya. After that, we will have opening remarks from Dr. Sri Pujianto SSc MSc as the head of the Biotechnology Study Program, followed by a group photo session, and then we will proceed to the main lecture, which will be delivered by Prof. Mario Konosawa, and then followed by a Q&A session, where you will have the opportunity to engage with our speaker directly. So the last agenda is closing remark. So before we go to the main event. As we open this event, let us honor our nation. So before we proceed, I kindly invite everyone to rise and join in singing our national anthem, Indonesia Raya. Thank you. You may now be seated there. Now, for the next session, I would like to invite Dr. Sri Pujianto, SSC MSc, as the head of the Biotechnology Study Program, to please come forward and deliver an opening remarks. To Dr. Sri Pujianto, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera buat kita semua. Dear Prof. Nuryo Kurosawa from Department of Science and Engineering for Sustainable Innovation, welcome to Diponegoro University. Welcome to our campus. Thank you for your time, for sharing your knowledge and experience for us, for my student, my lecturer. It is very important to upgrade my skill, my competence, especially for my student biotechnology program. Kepada para mahasiswa, jadi ini adalah kesempatan yang sangat-sangat berharga karena kita kedatangan tamu Prof. Nuryo Kurosawa ya, dari Soka University Jepang ya. Beliau adalah ahli mikrobiologi yang sangat terkemuka ya, terutama adalah tentang mikroba-mikroba Thermophile ya. Jadi mikroba-mikroba dari lingkungan ekstrim. Beliau banyak meneliti ya. Tadi saya berbincang dengan beliau, tempat mana yang paling ekstrim yang dikunjungi Prof. Beliau cerita di Antartika ya. Jadi beliau tinggal 2 bulan ya di sana ya, tumbuhnya Prof ya di Antartika. Jadi beliau sudah menjelajah kemana-mana di tempat-tempat ekstrim, mulai yang sangat dingin dan sangat panas, mencari mikroba-mikroba yang potensial untuk digunakan dalam industri ya. Jadi ini adalah kesempatan yang sangat bagus ya. Jadi memang Prodi Biotechnology, terutama Pak Anto sangat intens untuk menghadirkan para peneliti, para expertise dari berbagai negara yang kompeten. Prof. Nurul ini kalau kita lihat di Scopusnya, publikasinya sangat banyak ya, sekitar 88 paper, kemudian indeksnya 22 ya, saya aja baru 6 ini ya. Beliau sudah 22 ya, jadi citasinya sekitar 1.500, jadi sangat kapabel untuk kita serap ilmunya gitu ya. Jadi mohon untuk bisa dimanfaatkan sebanyak-banyaknya. Pada Pak Anto, terima kasih sudah banyak sekali memberikan kontribusi ke prodi ya melalui Reseach ya. Selamat mengikuti kuliah umum ya, semoga bisa mengambil manfaat yang sebesar-besarnya ya. Akhir kata, assalamualaikum warahmatullahi wabarakatuh, dan dengan izin Bapak Ibu sekalian ya, ibunya nggak ada, adik-adik sekalian, kuliah general lecture saya buka ya. Bismillahirrahmanirrahim.
Thank you so much, Dr. Sri Pujianto, SSC MSc, for this speech. So before we proceed to the next session, as to show our appreciation for Prof. Nuriel Rizal's time and invaluable knowledge, we would like to present a token of gratitude and appreciation. So I would like to invite Dr. Sri Pujianto, SSC MSc, to present the gift to the distinguished speaker. So everyone, please give a round of applause. So it's time for a group photo session first, because Dr. Sri Pujianto has another agenda. So everyone can stay on your seat and please rise.
Once more, 1, 2, 3.
So for the next session, let's move on to the highlight of today's event, which is the general lecture. But before we continue, let me first introduce our moderator today, Dr. Renat Antabudiharjo, SSA Biotech. Dr. Arna's educational background started with his bachelor degree in biology at Diponegoro University in 1991 to 1996, followed by master degree in biotechnology at Flinders University, Australia in 2001 to 2003, and doctorate in biotechnology at Hamburg University, Germany in 2006 to 2011. His research interests span from various topics such as PGPR and then thermophilic microorganisms and molecular biology. He also served as lecturers at Diponegoro University started from 1997 till present, and during that time he served as the head of Molecular and Applied Microbiology Laboratory at the Diponegoro University Center of Research and Services, and today he also serves as the head of the Research Collaboration Center for Thermophilic Enzymes. And so, without further ado, please welcome Dr. Renat Antoniadi, SSC BMN.
Oke. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Selamat datang semuanya. It's very honor for me to introduce to you our speakers today, Prof. Norio Kurosawa. So actually, I met with Prof. Norio last year when I was in Bogor attending college from BIN. And then I met with Prof. Norio. At that time we discussed, because we have a common interest in research object, which is thermophilic microorganism. And then we start to initiate collaboration. And I think this year we got the grant from your university for development of thermophilic microorganism. So, and luckily today, Prof. Norio can come to Undip. Actually, he just arrived today at 9 a.m. So I pick up him, and from Tokyo yesterday, only to meet you. Yeah, fly from away from Tokyo to meet you, to give general lecture and workshop. I think this afternoon and also tomorrow, and then benchmark in the afternoon. And after that, we will go to see geothermal site in Gedong Songo. So if anyone want to join with us, please. You can go with us, and then on Saturday, Prof. Norio will back to Japan again, to Tokyo. Okay, so before Prof. Norio give general lecture, I will read his curriculum vitae. Dr. Norio Kurosawa is now the Dean of Faculty of Science and Engineering, and the department is, it's quite long, Department of Science and Engineering for Sustainable Innovation. So it's kind of biotechnology things as well, yeah. So current position, Dean, Faculty of Science and Engineering, Sopra University, Professor since 2008, and no, the professor is from 2006, yeah, in Faculty of Science and Engineering. Research themes and experience: ecology of Southern Ocean, biogeography of coastal carbopods, taxonomy, physiology, and ecology of extreme thermophiles, genetic mutation, benthic fishes in Strait of Malacca, development of host vector system for hyperthermophilic archaea, and etc. and she got so many funding. The current ones is like exploration of engine of Thermophile for research and industry perspective. So I think there are a lot of research on extreme thermophile. And as you know, that Indonesia is very rich with geothermal site. So hopefully this general lecture will inspire you to explore our resources that can be used for industrial of biotechnology. So without further ado, please, Prof. Dorio, time is yours.
Good afternoon, everyone. So, nama saya Norio Kurosawa from Soka University, Japan. So, I'm very happy to get here. It's my first time to visit Smala. I have been Indonesia many places, but Smala is first time for me. So, Professor Pujianto, thank you very much for your kind introduction. And also, thank you very much, Professor Anto, for my introduction. And also, I thank you for you all for giving me a chance to talk here today. So, this is nice. So, hiding my note. Anyway. So let me start my talk. So the title is general lecture, but not so lecture. It's just talk. So I have been studying thermophiles over 30 years, very long. But I'm not sure I really like thermophiles, actually. Yeah. However, through my study on thermophiles, so I have been able to travel to many hot spring sites all over Japan, including deep sea hydrothermal vents, and also as well as to Yellowstone National Park in the United States, the volcanoes in the Kamchatka Peninsula, in Russia, and of course, just got to So later my research subject was expanded to various extreme files, not only thermophiles but also other kinds of extremophiles. So resulting in my going to Antarctica about 10 years before. So I stayed in the Antarctic continent for about 2 months. So it's very exciting experience. So today I want to talk about thermophiles and their proteins and some of my studies as examples of exploring thermophiles in the field work. So I said I'm not sure I really like thermophiles, but I'm interested in the origin of life. So in that sense, thermophiles, which are said to be the origin of life, are interesting subject, research subject. So next slide, please. So this is the evolutionary tree that includes all of life on Earth. As you may already know, so thermophiles are positioned near the root of tree. So this clearly shows that the last universal common ancestor, Luca, is the thermophiles. Of course, this Luca is not same as today's thermophiles, but it is certainly true that they are more similar to each other than to any other microorganisms. Therefore, studying thermophiles is no different from studying our original life. Next, I will talk about the definition of thermophiles. Next slide, please. Thank you. This graph shows the relationship between environmental temperature and the growth rate of living organisms. So temperature affects temperature affects microorganisms in two opposing ways. So as temperature rises, enzymatic reaction rate increase, so resulting that growth becomes faster. However, above certain temperature, proteins on the other cell components may be denatured or otherwise irreversibly damaged. So for every microorganism, there is a minimum temperature. Here, minimum temperature, below which growth is impossible, and optimal temperature, at which growth is most rapid, and maximum temperature here. So above which temperature no growth occur. These minimum temperature, optimum temperature, maximum temperature, sweet temperature called cardinal temperature of organisms. These characteristics of any given microorganism differ dramatically between the species. For example, some organisms have growth temperature optima near 0 degrees C, while for others, this can be higher than 100 degrees C. This figure shows the carbon and temperature curves for various microorganisms. In general, it is possible to distinguish four broad classes of microorganisms with their gross temperature optima. One is cyclophiles with low temperature optima. The mesophiles, like E. coli, with mid-range temperature optima. Many researchers define thermophiles with an optimum gross temperature of 50 degrees C or higher. In this video, thermophiles are further classified into three categories. One is ordinary thermophiles, and other is hyperthermophiles. Two categories. So, microorganisms most adapted to higher temperature are called hyperthermophiles. So, by the way, I have a friend who has been seeking the closest microorganisms to the origin of life. His name is Ken Takai. So he has found microorganisms maximally growing at 122 degrees C. So isolating from a deep sea hydrothermal vent. So this record has yet to be broken. This microorganism, called the scientific name is Thermophiles candelari, is an archaea that produces methane from hydrogen and carbon dioxide. They use the energy from the methanogenic process to synthesize organic matter from carbon dioxide. 4 billion years ago, the atmosphere contained a lot of hydrogen and carbon dioxide on Earth. So Thermophiles candelari, they have retained the characteristics of organisms that emerged in such an era. This Thermophilic archaea also has other interesting characteristics. Firstly, this microorganisms can only grow at temperature up to 116 degrees C under normal pressure. However, under 40 megapascal pressure, 40 megapascal means 400 atoms, so in a deep sea environment. So it can grow at temperature up to 122 degrees C. So, in other words, this microorganism is not only thermophiles but also Biotechnology microorganisms. More interestingly, so the methane produced by this archaea under high pressure is isotopically heavy. Do you know isotopically heavy? So normally it's difficult to explain in English. Sorry. So, anyway, normally isotopically heavy methane only produced by geochemically. So it was not believed to organisms produce heavy methane, isotopically heavy, but this organism produce isotopically heavy methane. So please study by yourselves. You don't know about isotopically, yes. By the way, do you know who discovered thermophiles historically? Somebody know? Who discovered? Okay. So it was 56 years ago. So it was discovered in Yellowstone National Park in the United States by Dr. Thomas Rock. So I want to share a video ended with this video. Yes, is described by Thomas Grock by himself. So, the last section of this video, he told about DNA polymerase, unstable DNA polymerase, and PCR purification of DNA. So, actually, Dr. Brock was just interested in unique microorganisms living in hot springs. So, many years later, so these microorganisms were applied to PCR, the evolution of molecular biology technique. So I think, as Dr. Brock said in the video, this is a typical example of how important basic research is. So if some of you are interested in basic research rather than application, so you may be asked, what is the use of this research sometimes? but you can just say with confidence that you are doing this because you like it. So next, I will talk about thermostability of protein of thermophiles. Ordinary proteins are very weak to heat, as you may already know. So proteins are made up of long chain of amino acid and have this kind of three-dimensional structure. When the temperature rises, this structure starts to break down and eventually stretch out. This is the thermal denaturation process. Once protein has been denatured, it is difficult to return to its original form, even when the temperature drops and it forms aggregates like this. So in fact, the proteins of thermophiles have a mechanism that prevents thermal denaturation. One of these mechanisms is called ion pair network. Please take a look at an actual example of the ion pair network. This is a protein called lucyl aminopeptidase from thermophilic bacteria, Thermophilus, and mesophilic bacteria, Lactobacillus casei. As you can see, they have very similar three-dimensional structures. However, the thermostability of these is very different. The thermophiles protein on the left does not denature even at 90 degrees C. So on the other hand, the mesophiles protein on the right being to denature at 40 degrees C. This difference is due to the difference in the number of interactions between the amino acid in the proteins. These figures are a enlarged view of the domain around alpha helices 1 and 2 of lucyl aminopeptidase from thymus and Lactobacillus. The blue parts indicates the amino group in the amino acid, and the red part indicates carboxylic group in the amino acid. The amino group is, as you know, positively charged, and the carboxylic group is negatively charged. When they are close together, they form an ionic bond, which are shown in dotted lines. Yeah, dotted lines. This is the interaction between the anion and cation. As you can see, the thermos protein has more ionic bonds than the Lactobacillus protein. The number is 11 and 4 respectively. These many ion pairs stabilize this helices domain in the thermos protein. The figure below are enlarged views of the domain around alpha helix 9. Also in this domain, thermos have 7 ion pairs, whereas the Lactobacillus has only 1 ion pair. This structure of ionic bonds is called an ion pair network. So thermos protein are stabilized by the presence of relatively large number of ion pairs. It should be noted that hydrophobic interactions are also strongly involved in the thermostabilization of protein. So I will not talk about the hydrophobic interactions today, but that is also important to protein thermostability, in addition to the ion pair network. So now I would like to move to the next subject and show some photographs of exploring thermophiles. First, I would like to show the nearest geothermal field to Tokyo. So it's Hakone Hot Springs. Sampling thermophiles usually begins with walking through these kind of steep places. So we can see it from distance, but hot water is gassing out between the rocks like this. We must check the temperature first, of course. So please take a moment to see a short video like that. The pH of the hot spring is also checked. Then once we have decided where to take the sample, we collect the hot spring water. We usually scoop up hot water and sediment in the glass vial. Can you see this small glass vial attached to the end of this kind of log? So actually, this log was made from kind of mop, cleaning mop. I detached the mop part and put some cleaning. So when the hot spring water is brought back to the laboratory, so a small amount is inoculated into the culture medium, and enrichment culture is started. So we usually use this kind of aluminum block heater and screw-capped glass test tubes for initial enrichment. If we are lucky, we can see some turbid cultures after several days to sometimes 1 month cultivation. Turbidity is measured using this kind of spectrometer. Once the turbid culture is obtained, we reveal the community structure of it by the 16S ribosomal DNA umbilical sequence analysis. Then, if we find a novel or interesting microbe in the enrichments, we then try to purify the novel or interesting microbe by dilution technique or single isolation on wet media. as shown in this slide. Through this kind of procedure, I first got novel species of Thermophilic archaea from Hakone hot springs many years before. So this archaea was named Thermophiles Sphaera and published in 1998. It grew optimally at 85 degrees C. That means this is hyperthermophile, and optimally at pH around 2. Starting with this species, we have discovered many novel species from Japanese hot springs. But important thing is, I also have wanted to investigate Indonesian hot springs. So because although Indonesia has many volcanoes, as you know, and consequently many hot springs, there have been very few appear. Indonesia has many hot springs, variety of hot springs, but there have been very few papers describing Indonesian thermophiles. So I really wanted to discover the Indonesian thermophiles. That opportunity came in 2015 thanks to the cooperation of IPD University, Professor Swan and Andy, PhD student at the time. On this occasion, we explored 5 hot springs in West Java. Today, I will talk about 2 of them. The first is Kawadomas hot springs. I think some of you may have been there. Somebody been there? Kawadomas. No. So, northwest from Bandung, I think. Northeast? I'm not sure. North from Bandung. So we sampled at 2 small hot ponds here. The first one is this. The first one is this. Yeah. So it was a hot pond under the large rock with a temperature of 65 degrees C and a pH of about 3. The volcanic gases were coming into the relatively clear. Oh, not this one. Sorry, I skip this one. This is the first small one. Temperature was 62 and pH is around 3. Yes, the volcanic gases are coming into the relatively clear hot spring water. So, very small. So of course we sampled the water excitedly with Andy, so wondering what kind of thermophiles so we could isolate. So but the result far exceeded our expectations. So we finally isolated an incredibly novel parasitic archaeal thermophile, Arma, from these hot springs. Do you know Arma? Have you heard about Arma? The Arma is the abbreviation of archaeal Richmond mined acidophilic nanoorganisms. It was discovered by metagenomic analysis and as an uncultured microorganism from acid mine drainage and reported by Baker et al. In 2006. So anyway, after this report, Baker et al. And other independent research groups have tried to capture these mysterious microorganisms. But nobody succeeded for over 10 years. So, by the way, someday, so Dr. Sakai, he was a PhD student in my laboratory, so told me that the almond-related DNA was detected in our enrichment culture. That was from Kawadomasan Street. So we excited and carefully checked the enrichment culture with microscope. This is a photograph of this enrichment culture. We could see very tiny cells and relatively large cells. So very tiny cells in like this. But they are larger than the viruses. So about 200 to 400 nanometers. So finally, we investigated that these tiny cells are almond, and the larger cells are the hosts are here. So these electron micrographs of almond isolate strain ARM1, isolated from Kawadoma's hot springs, and its host. As you can see, the ARM1 cells attach and sometimes seem to be fused with host cells. This isolate was published in 2022 as the first almond to be successfully cultured, purified, and characterized. Here is a D-pan in the title, but the D-pan is the name of the superphylum, which includes the almond groups. So the strain ARM1 is now, we deposited this archaea into the Indonesian culture collection in ACC and the Japanese culture collection GICM. So every researcher can use these microorganisms. Anyway, this video is shows interaction between the ARM1, strain ARM1 and host cell revealed by electron cryotomography. So we can observe needle or stool-like structures between the ARM1 and the host cells. So there are many connections between the almond and the host cell. So this is also published this year, and if you are interested in, please check this paper. This is the second pond in the Kawadomas. This pond was very close to the first pond, where the almond isolated. So only 3 meter distance. So, however, this pond seemed different from the first pond. It was muddy and unclear. temperature was 75 degrees C and a pH of about 4. So from this point, we also have isolated novel species of the genus Thermophilic. So please remember my first described species from Hakone Hot Springs. That was Thermophilic overquenches, published in 1998. For 20 years, the Thermophilic overquenches was alone in the genus Thermophilic, but his or her brother or sister was finally discovered in Indonesia. So this is the second species of the Thermophilic. So finally, I show the Kawakuti hot springs. So this is the gate of Kawakuti. So already evening, so actually gate was closed, but the officer very kindly give us the permission to enter the Kawakuti. Have somebody been there? Okay. Yeah. Yes, south of Bandung. It was very mysterious name by the fog and the dead trees. So it's very interesting. So it was already evening. It was already evening, so there was no one there except us. So I like these circumstances anyway. Then, of course, temperature checking. Well, temperature 33. 33? So, only 33. So I put my finger into the recorder, not hot. So I was a little disappointed because it was a long drive to get this place. So, but what pH? pH is about 0. 0 is almost same as 0.1 mole per liter hydrochloric acid. Very acidic. So we assume very interesting exophiles should inhibit this rate. So, however, so we got nothing, to tell the truth. We got nothing from this rate, even with relatively long time enrichment cultivation. So I thought there should be some microorganisms, but I don't know why we can get nothing. However, this is not a rare case. So I have experienced no growth many times so far. So it's usual. I don't remember why Andy and I took our photo here. Perhaps it was because we felt so maybe we are a good match for this experience. By the way, anyway, this is our last slide. So I want to try the enrichment cultivation again. So I want to go Kawaguchi again and try enrichment to get super extreme profile. Not thermophiles, but very acidic feeling. Okay, this is the end of my slide. So, if some of you are interested in extreme files, especially thermophiles, so please join to the Grove Anton's group to explore thermophiles in the field. It's very enjoyable. Thank you very much for your attention.
Okay, thank you very much, Prof. Nurio, for your sharing regarding the extreme thermophile microorganism. So as you see that actually Indonesia is very rich in geothermal site, and every time if you want to explore the microorganism, you get something new from there. And in Indonesia we have a lot of geothermal sites. So this site is ready for you to be explored. So now it's depend on you whether you want to explore this richness of Indonesian resources or you just leave it like that. Yeah. So now we go to discussion. So please, if you have any question, feel free to ask to Prof. Nurio. Ya, silakan kalau ada yang mau ditanyakan. Okay. State your name, ya.
Excuse me, Prof. My name is Louis from Biotechnology student. I wanna ask. I'm very curious about how can thermophilic microbes destroy or kill apart from the lack of nutrition or the environmental temperature dropping drastically. Thank you.
The question is if the temperature is so down, so how can they survive? So I think some of them are dead, but some of them survive in a spore-like structure sometimes, and the micro-thermophiles which cannot make spores, so just sleeping. Yes. But it's very interesting. So how can they survive? Because not only temperature dropping, sometimes the water gone and dry out. So, but they always recover. So it's very interesting. So it's a very good team of research, I think. Nobody knows.
Okay, next question.
Okay, my name is Angel. I want to ask, I think, 2 questions. My first question is, what properties from mesophiles and thermophiles makes them different? And relating to the first question, because we are in a global warming era, what can global warming make more thermophiles, and can mesophiles become thermophiles because of adaptation? Thank you.
So actually, thermophiles thermophiles and mesophiles are different in the point of protein stability. So because most microorganisms uses other small compounds in their cells, like ATP are same. So lipids are almost same. But I think just protein is very much different between them. So, in the global warming era, so I think the thermophiles are living at least 50 degrees centigrade and higher. So, but global warming is maybe 2 or 2. I'm not sure. So, how temperature rise in the future, but I think it's not. It will not affect to the composition of the mesophiles and the thermophiles.
Maybe we ask, is there a possibility that mesophile and bacteria can turn to like thermophile to this global warming?
I don't think so. Because, as I mentioned, thermophiles define grow at least 50 degrees C.
So at least 50 degree. Ya, jadi thermophile at least di atas 50 derajat. Di bawah itu misalkan 45 termotoleran, belum masuk ke thermophile. Ya. Jadi batasnya itu di atas 50-an itu. Oke. Oke, next question. Oke.
Okay, thank you for the presentation, Professor. I want to ask something. In ARM, what is the symbiote of Metallospira? What is the function of symbiosis between the ARM1 and Metallospira?
You asked about what for the interaction between the arm1 and the Metallospira. So the stolen arm1 cannot survive without host cell. So absolutely necessary the presence of host archaea. And once arm1 infected to the Metallospira, so Metallospira growth is down. So that means they are not helping each other. So the arm1 is parasite to the Metallospira. That is not fair symbiotic relationship. It's not fair. So the arm1 has incomplete genome. So for example, TCA cycle is incomplete, and other amino acid synthesis, some of our amino acid synthesis is incomplete pathway. So I think the arm1 get amino acid or some small compound from the host cell.
Mr. Devani? Not yet? Maybe you have to read the paper that has been published so you can understand more. Yeah. Okay, any other question? Okay, silakan.
Hello, my name is Amalena Widaramani. thank you for the opportunity for asking you. I'm curious about the microbes that you've included, which is can produce sulfur. Have you ever analyzed how those microbes can actually produce the sulfur? Like, what is the difference of their metabolism system with the other microbes that mostly are actually produce organic compounds? Thank you.
Today I have not talked about the diversity of thermophiles, but actually there are many kinds of thermophiles. Some of them are heterotrophic, that means they require organic compounds, but others are really chemonisotrophic. So that means they can produce organic compounds from carbon dioxide. Very rare, there are much kinds of thermophiles. So it's not difficult to say this is one. So sulfur. So in the sulfur effect, some thermophiles like sulfur. For example, the Sulfurispera ovocensis and the Sulfurispera javensis isolated in my laboratory. So they very much like sulfur. So elemental sulfur. They oxidize sulfur to get energy. So that the name of the scientific name is derived from sulfur.
Okay, another question. So this is your chance to really asking about this very interesting microorganism. And like I told you before, Indonesia is very rich in geothermal site. And in fact, Indonesia is one of the countries which has the most volcanoes in the world, right? We have around 144 active volcanoes. So right now you can see that there is a volcano eruption in Wakatobi or Gunung apa ya, yang di NTT. So there is an eruption right now in the volcano in one of Indonesia island. So that's why even in Semarang we have many geothermal sites, like in Gudoharjo, in Mijen, and also Gedong Songo, this kind of geothermal site as well. And tomorrow afternoon, me and Prof. Norio, we will visit Gedong Songo. So if anyone would like to join, just go with us. So we can go together.
And you mentioned before, so there is a very salty hot spring, right?
Yeah, that's mud volcano. Mud volcano actually, for my student here, they isolate bacteria from mud volcano. That's in Bleduk Kesongo. The pH is 9. The pH is 9 and the salinity is 4%. So means that 4% is higher than even from the water sea, because the water sea is only 2 under 3, but this mud volcano is 4. Yeah, so it's quite interesting.
So maybe so not only thermophilic but also resistant to saline.
Yes. Yeah.
So very interesting for industrial application.
Yes. Yeah, some of the students already like isolate the enzyme. Apa enzimnya? Apa? Protease and amylase from this glutathione. Yeah, and they already have sequence the 16S and materinya apa? Aluminas, dan oke. Coba kalian pakai ini.
Hidrantia bukan? Hidrantia.
Hidrantia and aluminas. Yeah. So based on 16S, identification, the isolate is aluminas and the.
Okay. And you are characterizing the proteins from
Okay, the test screen. Yeah, the test screen. Maybe will be continued with the protein characterization. Ya, Patan mungkin kamu bisa menggunakan itu buat risetmu later on. Okay. So, any other question? Silakan kalau yang mungkin pengalamannya Prof. Nerul waktu di Antartika. Okay.
So, thank you Prof for the interesting presentation. So I would like to ask about, I'm curious about the thing that you say when you want to sample at Kawah Putih, where the condition is very acidic. So how do you plan to store the sample before the analysis on laboratory? Is it do you need like a special vessel to contain the sample? Because or is there any possibility for the microorganism or the sample to die because of the fluctuation of pH if it's not maintained before the maybe like cultured in laboratory? Maybe that's the question. Thank you very much.
Yes, I'm planning to use a wide variety of media at first and variety of culture condition, for temperature and pH, and I think actually I saw some green mud in this land, so maybe some kind of very acidophilic cyanobacteria, for example, is present. So I also plan to use the light to when I enrichment culture. So blah blah.
But how do you create to the laboratory?
Bring back.
How do you carry from the site to the laboratory? You just use like ice block box? Or
No. So I usually transfer the sample in ordinary temperature. So no temperature control. Many years before, I was also wondering about that. So I use pot. So, but it's not much effective to survive the microorganisms. Up to 1 week, I think. I have not checked longer than 1 week. And if possible, so it will require a long time to bring back to Japan, so it's better way that I will cultivate in his laboratory. Initial enrichment time.
Oke, ada lagi? Silakan.
Thank you for the opportunity. Good afternoon, Prof. I don't get any question about thermophiles, microorganisms, but I just want to know about your stories in Antarctica. I mean, I'm curious about what you're doing in Antarctica and what organism that you find there, or maybe anything different about the Thermophiles microorganisms and the organisms that you found in Antarctica.
The Microorganism, microorganism.
Yes. In order to find novel microorganisms that adapt to low temperature, that's the reason why I tried to go to Antarctica. So after about 10 years, my study on Thermophiles, so I was also interested in opposing microbes to the Thermophiles. So what kind of protein they have or something like that. So, and also I just wanted to go to Antarctica. So microbe, the study, research microbe is very good for traveling everywhere. So because the normal animals and plants cannot survive in hot spring or Antarctica, so but microbes are everywhere. So if you will be a microbiologist, so you will have a chance to go to everywhere, including Antarctica and Arctica and the deep sea.
Okay, one more question. If you have any question. Okay, so I think that's all for general lecture today. Thank you very much, Prof. Rio. We give big applause for Professor Rio. Thank you very much. You come to Semarang directly from Tokyo just to meet the student, and then going to sampling, and Saturday back again to Tokyo. We really appreciate that for your effort to come here. So hopefully we can continue our collaboration. So student, if you interested in research on thermophilic microorganisms, there is possibility that you can go to Prof. Norio lab in the future if we can get you a grant for the research internship. So you can stay in Prof. Norio lab for a couple months working there. Next year on March, I will be leaving to Prof. Norio lab for a couple of weeks to continue our collaboration. Hopefully, I can get many interesting microorganisms. And so, if you really do want to explore the geothermal side of Indonesia, so yeah, feel free to join with the lab in UPT Terpadu, because we already have research collaboration center in Thermophilic enzyme, where we have collaboration with UGM, BRIN, UTM, and SCU on this specific topic. Yeah. Okay, so Prof. Norio, once again, thank you very much. We presented that so that the student can have more information on microbiodiversity. Thank you very much.
Yeah, thank you to Professor and all of you. So it's very nice time for me. Thank you very much.
So thank you, Dr. Anto, for moderating the session, and thank you very much, Prof. Norio Kurosawa, for such an insightful and inspiring presentation. Maybe your very exciting journey for exploring thermophilic microorganisms have truly deepened our understanding of this fascinating field. And I'm sure everyone here has gained valuable knowledge that will spark new ideas and curiosity in the future. So thank you very much for all of the participants for the engaging discussion, and thank you for everyone who actively participated. That brings us to the end of today's general lecture, and on behalf of the organizing committee, I would like to express our gratitude once again for the valuable insight and to all of you for your active participation. We hope you found today's session enriching and inspiring. Thank you, and we look forward to seeing you at the next event. Wassalamualaikum warahmatullahi wabarakatuh, and I wish you all have a wonderful afternoon. Bisa inisiasi terlebih dahulu ya.
Ya, oke, ininya bos.